quick overview of compartmental modeling. I've already given you an intuitive description of compartmental modeling. We will take a dendritic cylinder and break it into small sub-chunks and write governing ordinary differential equations for the dynamics of each chunk and then bring them all together and numerically integrate them simultaneously. It's a different approach than what Will Rawl took. He took a more mathematical pencil and paper approach. This was because it was in the early 1960s. Compartmental models are based on data like these. I've shown you this example before. It's an example of how individual neurons' dendritic architecture can be very, very, very precisely constructed at high resolution, in this case using 2-photon imaging. And so the approach will be to collect images such as these and perhaps break this chunk of the dendrite into a small tube. We assume that compartment is isopotential, and we'll do that throughout the dendritic tree, possibly many thousands of compartments, and write governing ordinary differential equations for the time rate of change of voltage within the dendrite in response to something like a current stimulus applied somewhere else in the dendrite. So this is how that problem goes. We're going to start off with a simple cable structure, as we always do. You saw a diagram like this when we derived the cable equation, but now we're going to use this diagram, which is, we can think of this set of electrical circuit components as representing an individual compartment. So this is the axial resistant for the jth compartment, and this combined electrical circuit element here is actually a capacitor in series with a resistor. The capacitor represents the total membrane capacitance of that small sub-chunk of cylinder, that compartment, and the resistor represents the resistance to current flow out through the membrane of that compartment. So this is axial current flow, this is transmembrane current flow in the different compartments. So pick a compartment, compartment J. We know that the current flowing into node J is equal to the potential at node J minus 1 minus the potential at node J times this conductance, that's the current through that resistor slash conductor. So that's this entering current. We know the current entering from node J plus 1 is the potential at node J plus 1 minus the potential at node J times this conductance value. So we've got this current plus this current directed into node J must equal the sum of the other currents entering the node. So we have a current entering the node that we can consider to be an applied current. That's the stimulus. And the current leaving node J is that current through these two circuit elements. And so there are two components of that, right? So there's a capacitive current component, and there's a current through the membrane resistance. And this E sub r here, we're going to think of the membrane resistance as in node J as being denoted by this symbol here, and that when that conductance, that conductance is selectively permeable to some ionic species with reversal potential ER. So that's how ER gets into this equation. Whatever the reversal potential is that controls flux of charge through the membrane, the difference between that reversal potential and potential at node J is the electrical driving force through the resistive circuit component of the transmembrane impedance. So these kinds, this equation, if you are with me, if not, raise your hand. This equation applies to all of the interior nodes. Let's assume this was a finite length cable. So there's an ending or capping node at one end, and there's an end node at the other end. And so if there are k nodes altogether, k compartments, this equation applies for all of the interior nodes. And there'll be a slightly different equation that we apply at the beginning of the cable and at the end of the cable, because there will be an axial current component missing at those two locations, because the cable starts and ends at those locations. But fundamentally, all of these equations come from the same considerations. And if we break a dendritic branch into k compartments, we'll have k such equations, right? And so they're going to be linear ordinary differential equations in terms of voltage, so we'll have derivatives of voltage. So our goal is to isolate the derivative, the time rate of change of voltage on one side and put all of the other terms on the other side. That's the velocity field. So we'll have a system of coupled ordinary differential equations for voltage within each little compartment from which the cable is composed. And when we do that algebra, when we isolate out the dV dt's on the left-hand side, we'll get equations of this form on the right-hand side. This is known as the velocity field. So it's easy to write down those terms. Okay. And so now what I want to do is to just rewrite those equations in matrix form as a way of showing you that this problem is really a problem that involves a linear, constant parameter multivariate system. And so we're going to put these equations into the kind of form that one uses when thinking of dynamical systems and control theory applied in linear systems. So we're going to write down the state equations. So let's define a vector V that has as elements voltage in each of the k nodes. So V's a column vector, and so it's equal to this. This is the transpose operator. So that's how we define V. We're going to define a vector that we consider to be inputs. We're going to call it U of t. And U of t is the applied stimulus current at each node. It is an input. That's what we're thinking of as the input to the system. So there are k nodes, k possible values of I. Typically, we're interested in what pattern of voltage a current injected at one location in the dendritic tree will be produced. So typically, many of these terms are zero, and there's one that's non-zero. We're going to define a matrix B that is a diagonal element, a diagonal matrix. So that's how I write it, as a diagonal matrix, with the diagonal matrix being 1 over the various capacitive terms, one for each of the compartments. And then the most important matrix here is what we'll see is eventually known as the state transition matrix. It's the matrix A, and we're going to define it as so. So this rho sub 1 here, this term, is equal to, for an interior node, the two connected axial conductances plus the transmembrane conductance. To get these matrices and vectors, all you need to do is to go back to the last slide and look at the form of the system of coupled ordinary differential equations and arrange terms as I've shown here. And when you do that, the point is that the state equations can be written in matrix form, and they look like this. So time rate of change of voltage, and I've normalized voltage in a way that I haven't described here. This is for a normalized version of the voltage, is equal to the matrix A times voltage plus the matrix B times the inputs U. Remember, U is a vector of current inputs. So you're familiar with equations of this form from signals and controls. This is a state transition matrix. It has constant parameters, so whatever the axial conductances are, and the membrane capacitances are, and the transmembrane resistance, they get packed into A, the matrix A. We'll consider the form, the structure of A in just a moment. And B is I think we said it was a diagonal matrix, right? Yeah, it's this diagonal matrix here with terms 1 over the capacitances associated with each compartment. So these equations here can be written in this form. So this is a time invariant linear dynamical system of equations. Now, if we are writing these equations for a cable, then A takes on a special form, an unbranched cable. A will be what is called a triadiagonal matrix. There will be elements along the diagonal, and there will be elements off the diagonal, but every other element of A is 0. Intuitively, you can see why, because any node is interconnected to only one node on the left and one node on the right, one compartment on the left, one compartment on the right. So that sets up the tridiagonal nature of the state transition matrix A if we are developing a compartmental model of an unbranched cable, an unbranched dendrite. So there A has a special form. But in general, neurons, dendrites obviously branch. The one I showed you there branched. And so depending on how we number the compartments, the elements of A will get filled in. But in general, there are not many elements of A to filled in. It's what's known as a sparse matrix. It's got a few elements, but not all that many. So there's a lot of non-zero elements. Not every compartment connects to every other compartment, obviously. Every compartment only connects to a handful of other compartments. And so A is in general a sparse matrix. And so there are lots of open source software applications for solving these kinds of problems, for solving for V, given knowledge of the matrices A and B and some particular inputs. So MATLAB can do this. What is it, ODE 45, something like that. It's a common function that is used in MATLAB to solve these systems. There are many other open source numerical integration packages that can solve this kind of problem. So if I were to give you a neuron, I wish I had done this for a homework, give you a neuron and some compartmental decomposition of that neuron and asked you to simulate its electrical response to a current applied somewhere in the dendritic tree, you'd work out what the matrix A is, what the matrix B is, where that input is applied, what's its index, and then you could use a numerical integration package like ODE 45 in MATLAB to numerically solve this equation for voltage as a function of time in response to any input in the dendritic tree. Now, we're dealing with simple circuit elements in our models. I haven't said a word about voltage-dependent conductances. They can all be incorporated into this kind of formulation. It's just that wherever you see a transmembrane conductance, that conductance, mathematical description of that conductance is now going to be more complex. And for voltage-dependent membrane conductances, they will depend on voltage. So the state variable V will now be an element in the state transition matrix A, and that makes the system nonlinear. And so it's not a system that we can solve even for cables with pencil and paper anymore. But we can still use the numerical solvers in MATLAB and other applications to solve for V, even when we make the transmembrane conductances very complex. And then there are specialized applications like Neuron, which is an open source tool out there on the web for doing neuronal simulations. You give Neuron a list of compartments. I think it could be as little as simply the dimensions of the compartments. What's the diameter? What's the length of each compartment? And what are specific resistivities, the R sub i's, the R sub m's? What's the specific capacitance? And Neuron will build the model and do the simulation that you want for you. So that's a quick overview of how you generate the governing equations for compartmental models. We solve them using numerical methods. And so another important aspect of this is, what if we go back to these equations here and we set this left-hand side to 0, and in so doing, dv dt is 0 everywhere now. And so the v's that solve that equation will now be steady-state solutions. So when we apply a current and maintain that current, what's the steady-state distribution of voltage in the dendritic tree? Well, to figure out what it is, just set the left-hand side to 0 and solve for V. And so V in the steady state is just going to be A inverse times B. And so we have a voltage here on this side. The elements of U are current. And so we're just applying Ohm's law, right? This is Ohm's law. E equals I times R. A inverse times B, that matrix, is a matrix of resistive elements. And the elements are known as transfer resistances. And so from A inverse B, the elements, Kij, tell us how a current applied at one location I maps to a voltage at location J. So we can deduce these things called transfer resistances from knowing what A inverse times B is. The elements are transfer resistances that allow us to understand how a current applied out at the dendritic tip of the translates to a voltage generated somewhere else in the neuron. And we'll use this concept of transfer resistances elsewhere in this lecture. That was a rapid overview of compartmental modeling. It's just what it says. Break the neuron into compartments, solve coupled systems of ordinary differential equations for voltages. Compartmental modeling. Now we're going to think about signal processing in dendritic trees. And the first signal processing strategy that came out of Will Rawls' work, but as it was applied by two very famous computational neuroscientists. And they still are in the game. They're still very famous. There's Tommaso Poggio at MIT. There's Christoph Koch at Caltech. So Koch and Poggio really picked up on the work of Will Rawls, and they were really the people that in a big way took that theory and applied it to understanding the kinds of calculations that can be done in dendritic trees. And I was very influenced by their work when I was a graduate student. I thought it was terrific. I still do. And so one of the first things they considered was the simple notion of dendritic integration. When synapses land within a dendrite, what they showed was how the co-localization of those synapses can limit neuronal response to electrical activity in each of those synapses, the activation of each of those synapses. Whereas distributing those synapses across the dendritic tree really amplifies response. It's an example of what is called dendritic integration. And so I want to explain how that comes about because it gives us insight into a design strategy. If you have a set of inputs to a neuron and you really want to amplify that neuron's response to those inputs, distribute those inputs into electrically distinct regions of the dendritic tree. That's when you'll get the biggest response. Now, how does that come about? So to understand that, we're going to go back to the simple single dendrite neuron model. And this time, we're going to switch the way that we think about what an input is to a neuron. In the Rawls world of linear systems, we thought of inputs as being injected currents. We're changing that view now. We're now going to treat the input as a synaptic conductance change because that's what happens in a neuron when a synapse is activated, a neurotransmitter is released, and it binds to receptors in the neuronal dendritic membrane. The binding of that neurotransmitter increases membrane conductance to some ion or some family of ions. And there's a reversal potential associated with the complement of ions to which the membrane channel becomes permeable to. So that's represented here as E, and the conductance change here is G. All right? So whatever the voltage is inside the neuron, let's say it's V, this current is now E minus V times G. We're going to think of G as the input now. G, a conductance change, is the input, and the current that flows into the dendrite is determined by this reversal potential and the potential inside the neuron. It's determined by their difference times the conductance value. So normally in the absence of synaptic input, we're going to assume this conductance value is 0. An action potential comes down, the projection of the source neuron causes neurotransmitter release, binding of neurotransmitter, that increases membrane permeability. That's the input. Now we're going to ask the question. Suppose you've got a bunch of inputs, bunch of synapses, and you concentrate them at the same place. The fact that you've localized all of these synaptic conductance changes, what does that mean about the nature of signal processing in that neuron versus a situation when you would take that same total synaptic conductance but just distribute it in a different way across the dendritic tree? There can be a huge difference. And this illustrates what the huge difference is. So, a synaptic event, the act of binding a neurotransmitter somewhere in the dendritic tree at a synapse, increases this G. It's not 0 anymore. Now G is non-zero. And the electrical driving force that is moving charge into the neuron from our prior diagram is E minus V sub I. V sub I is the location that the synapse is at. So that's the current generated by activating this synapse. This is the current that's flowing into the neuronal dendrite at the site of the synapse. Okay? So the voltage where this event occurs is equal to the current that's flowing in through the synaptic conductance at that site times one of these special transfer resistances. And I'm denoting it as KII. KII, so I indexes location, this is where the synapse is. So this is the input resistance of the neuron at the site where the synapse lands. Picture you should have in your mind is this neuron is made up of a branched cable conductor. That is the dendrite, and there's a soma attached to it somewhere out there in the dendrite. There's some input resistance, input conductance, looking into that neuronal structure at the site of where the synapse is located. That's what KII is. It's the input resistance. And obviously, the current applied at a point times the input resistance at that point is the voltage at that point. How do you get KII? You go back to those equations that I just derived in the steady state. KII are going to be the diagonal elements of A inverse times B, that matrix product. Those are these input resistances at each of the compartments. So you pick out the right one corresponding to the compartment where your synapse is. That's the KII value to use here. Does that make sense? Yep, okay, good. So let's just plug things in and isolate terms. So we've got an expression for the injected current. It involves V sub I. V sub I is on the left hand too. So if we isolate V sub I, we get this relationship for the local potential. As simple as it is, this relationship tells us something very important. Imagine letting G become very large. This neurotransmitter binds to a receptor and the change in membrane conductance is huge, huge conductance value. Well, this term is going to something near 1 when that happens. And this term can't be it's bounded from above by 1. So what does that mean? It means that the local potential produced by even a massive conductance change applied at one point is never going to be bigger than the reversal potential. And so this fundamentally is an example of a nonlinearity. There's a nonlinear relationship between membrane conductance and the voltage that it produces at the site it applies. That membrane, that voltage saturates. It's just intuitive. You think back to the circuit diagram. Whoops, I'm going the wrong way. Here's the circuit diagram. G goes to infinity. This node can't be tied to anything other than the value of the battery E, the reversal potential. So that's a kind of nonlinearity. We can't induce a larger voltage change in that value of E. Whereas when we treat current injected current as a description of what a synapse does, making that, we can make that current as large as we want, and we'll get a bigger and bigger voltage. That's one way in which that scenario differs from what real neurons do. There's this thing called synaptic saturation, and that's what it is. So let's think about how this voltage then maps to voltage at the soma. So the voltage at the soma is going to be equal to the current that enters at node I times the transfer resistance between node I and the soma, which is indicated here by the subscript S. So this is the transfer resistance between wherever node I is and the soma. By definition, that transfer resistance is how current at location I maps to voltage at the soma. And so VI over VS is just KII over KIS, the current sources divide out here. And so voltage at the soma can be written as KIS over KII times the voltage out there in the dendritic tree. And if we bring this term over, then we've got an expression for voltage at the soma. So what this expression says is, yes, voltage out in the dendritic tree produced by even a large conductance change is capped at E, and the voltage produced at the soma is also capped. There's a maximum value it can produce, and it's E times this quantity here. Remember, this is going to 1 as G goes big. So it's this quantity here. And what do you think? Why is KIS over KII always less than 1? Why is that true? KIS is always less than KII. Why is that true? There's a simple intuitive explanation. It's saying the input resistance, looking into where the synapse is located, is always lower than the transfer resistance between that location and the soma. How do you get that intuitively? Yeah?
I mean, there's just a greater distance to travel, so there's more resistivity across, like, greater distance.
Not greater resistivity, because we said that KIS is less than KII. So it's actually less, the transfer resistance between your location and the soma is always less than the input resistance at your location. But it does have to do with the distance. That's a part of it. So when charge moves from the location where it enters the dendrite to the soma, there's a lot of pathways by which that charge can flow to ground. It can flow out through the membrane resistance, so you lose more charge. And so that corresponds to lesser resistance to charge movement to ground. So this term is always less than this term. And so the voltage that you can produce at the soma, the maximal response, is given by the maximal response that you produce out there in the dendrite times the ratio of these transfer resistances. So this is all known as synaptic saturation. Now let's see what it looks like. So here's a plot of the log of G times the input resistance and the value of this function as we vary that log. So when G times KII is 10, the value is of this function is about 0.9, and when I can't see it, it's whatever it is, it's about 0.9. And when G times KII is 100, the value of this term is very, very close to 1. So what does this mean? Why am I harping on it? Okay, let's take the example where G times KII is 100. So whatever the conductance value is large, and this product is 100. Voltage at the soma is about this. Suppose we reduce this G by a factor of 10. Okay, well then the voltage at the soma, we can pluck off the value of this function for an argument of 10. It's about 0.9. So the voltage at the soma is still 0.9 times this stuff. So we reduced the value of the synaptic input by a factor of 10. and we're still almost saturating our voltage response. So now let's imagine, we're not going to derive this, imagine taking that reduced conductance value, it's G over 10, and distributing it in the dendrite at electrically distinct regions. So that and that means that these regions are far enough apart electrically so that the conductance changed applied over here doesn't have any effect on the response to the conductance change at this part of the dendritic tree. Under those conditions, the currents that we produce flowing into where the synapses are located, they're going to sum up at the soma. And if we're applying this G over 10 conductance value at 10 different locations, we're going to amplify response at the soma by about a factor of 10. So we're going to get 9 times this value at the soma. Whereas if we took those 10 conductance values, each of magnitude G over 10, and applied that lumped conductance, which is just G, at one location, we get a much smaller response at the soma, 10 times smaller. So this is the significance of synaptic saturation. It's what's causing neurons to do what is called dendritic integration, the summing up of inputs, much more effectively when inputs are distributed spatially to electrically distinct regions of the dendrite. And you can get a very considerable amplification of response when you do this. So it's a design strategy. If rather than put one big synapse on a dendrite at one location, if you want to make that neuron fire reliably, distribute that synaptic conductance change around the dendritic tree to electrically distinct regions of the dendritic tree. Then you can most effectively stimulate the target neuron. So distributing synaptic inputs contributes to dendritic integration and the more effective driving of the target neuron. If it's important that a signal be reliably transmitted, that every time an action potential is generated in a presynaptic neuron, you want to make sure that postsynaptic neuron fires. One strategy for doing that is to apply multiple inputs at distinct locations in the dendritic tree of the target neuron. It will tend to fire far more reliably. There are many scenarios in neural signal processing where the preservation of temporal information is important from stage to stage as we transmit signals through neural networks. And so this is one strategy for maintaining temporal coherence. That's the importance, in my mind, of this piece of work. And one can prove this result. One can mathematically prove it. We aren't going to do that, but it can be done. So here's an example. This is from the work of Koch and Poggio. They looked at retinal ganglion cells. The advantage of working with these cells is that since they're in the retina, and the retina is a two-dimensional object, really, it's an array, these neurons tend to have flat dendritic trees. And so when they looked at them under the microscope, they're projecting onto the tracing paper where they're drawing out the dendritic tree, they're projecting an object that's flat. And so they can trace it reliably and get some good measurements by using this technique of tracing. So that's what they did, and they built electrical models as we have described for these dendrites. And here's a kind of retinal ganglion cell, an alpha ganglion cell, whose dendritic tree they found a special property. We won't go through the strategy by which they did this, but I think you can now understand it intuitively. They were able to identify electrically distinct regions within this dendritic tree. Events here have little influence on events here, and events here, and events here. That's the meaning of electrically distinct regions. And so this is an example of a neuron that has a dendritic tree with enough subregions in it that it can effectively do dendritic integration. Whereas, here's another kind of retinal ganglion cell, a beta ganglion cell, that has different receptive field properties measured psychophysically. And notice that this is a 100 micron scale bar, so the whole dendritic tree is about 100 microns in diameter, and that's the same scale bar here, so these are much, much bigger neurons. So for these beta ganglion cells, being physically smaller, they weren't able to identify large, distinct subregions of the dendritic tree. This neuron is, in effect, less well suited for performing the function of dendritic integration. You put an input here, it's not so different from putting an input there. They're kind of in the same place electrically. And so the synaptic saturation property that I described, it's more likely to happen in this neuron. This neuron is going to integrate signals at different locations in the dendritic tree less effectively than the neuron that you just saw, the alpha ganglion cell. So this just summarizes that. I've already said it, so let's move on to the next topic. I think this is pretty interesting. Here's another example of an important computation. Sometimes it's called coincidence detection. Koch and Poggio referred to it as a way that neurons can calculate an analog AND operator. They can implement the AND function. And here's why it's important. In audition, and other things, but great example in audition. Okay. Somebody over here, Dr. Winslow, stop. I turn in that direction. Why do I turn in that direction? Because the sound reaches this ear. Before it reaches this ear, there's an interoral time delay. And there are central auditory neurons that are very, very finely tuned to specific interoral time delays. And its interoral time delay is the cue that we use to localize sound in the horizontal plane. So I know to turn over here because there's a neuron or a group of neurons in my medial superior olive at a particular location in my medial superior olive that are lighting up, and they're sensitive to a particular interoral time delay, so I know somebody's talking over here. This has been studied in barn owls. Barn owls have an exquisite ability to sound localize, and so the neural circuitry underlying sound localization has been heavily studied in barn owl. We know that the neurons in the mammalian medial superior olive and in, it's in birds, it's called nucleus magnocellularis or something like that. Anyway, they all have the same structure. They're bipolar neurons. Here's one of them. So there's a cell body and always two branches coming off the cell body. Ipsilateral inputs from the same ear go to one side exclusively. Inputs from another ear go to another side exclusively. And these neurons respond preferentially to sound at a particular angular location in the horizontal plane. So the question is, how do they do this? It's an example of an AND operator. I'd rather return to this in a moment. I'd rather go to this movie. There was a hypothesis put forth a long time ago. It's called the coincidence model. It's proven to be correct. It's been demonstrated computationally. It's been demonstrated experimentally to be correct in an essential way. So let's say a sound source is over here. That sound arrives at the cochlea on this side of the head before it arrives at the cochlea on that side of the head. We're considering an array of neurons. This happens to be the medial superior olive on the other side of the head, right? There's two of these structures, one on each side. So it takes electrical activity in these primary auditory nerve fibers longer to propagate over to the contralateral side. And there's also an interoral time delay that determines the propagation time required for a sound impinging on our head here to wrap around the head and hit this cochlea, and the path length to these neurons in this medial superior olive is shorter than this particular path length. So this is just describing the anatomy. So assume there were no difference in these path lengths. Assume they were the same. And assume that the sound is directly in front of us. Then the sound would hit this cochlea and this cochlea at the same time. The sound would activate primary auditory nerve fibers. It happens to be in a stochastic way, but on bulk, the electrical activity in these projection axons would reach this neuron at the same time, and this neuron, and this sound would reach this neuron at the same time. And so both the electrical activity from these neural inputs would be coincident at this neuron if there were no difference in path length and if the sound was right in front of us. Now, let's assume the sound is in front of us, but there is a difference in path length. It takes it longer for the sound to arrive here. Oh, I didn't say that right. I missed something very important. I said that wrong. This is very important. Notice that there's a difference in how these primary auditory nerve fibers project. On one side, they're projecting to this neuron, and then they send other projections to these other neurons. The pattern is reversed on the other side. The first neuron activated will be this one when sound reaches here. And the last one activated from this side will be this one. Scratch what I just said, delete it, I was wrong. When this path length is the same as this path length, the sound doesn't arrive at that neuron simultaneously. What will happen is the sound from this side arrives at this neuron at the same time that the sound that propagates from this side arrives at this neuron. Then they'll arrive at these two neurons at the same time, these two neurons at the same time, and they'll arrive together at this central neuron. That's what I should have said. I blew it the first time. Okay? So now, let's change this delay line. So, by making this transmission path longer, this electrical activity will propagate to and impinge on more and more of these neurons before this input gets there. So if the sound is directly in front of us, if this path length is longer than that one of these neurons will be the one that the sound-induced electrical activity arrives at in a coincidence way. So I hope I said that clearly. Let's just watch the movie and make up for whatever I didn't say clearly. All right, move going. Sound is right in front of us. Gets here first. Okay, that's the neuron where the coincident electrical activity landed. We're repeating it. Electrical activity arrives here first, and then progressively, but that's the one where the sound, the electrical activity is coincident and we get a response, okay? So now the next part of the movie is the sound comes in from this side. Now watch what happens to the site of coincident input. It's shifted to down here. And that's because this side gets activated first. So these signals are arriving more quickly than they would otherwise along this neuronal array. Yes?
Is there a way for the first one to go true, and then by the time the second one gets activated, they're all gone true, so nothing?
You can't do intraoral detection? And that would be the consequence, that there's a set of intraoral time delays that you can't detect. There's a way for it to happen. It's not a biologically valuable way for it to happen. It's a biologically detrimental way. And that's probably very significant. I don't say that flippantly. I think sound localization is imperative for the survival of an organism. It's how you know that there's a grizzly bear over there. That's one way you know that there's a grizzly bear over there. And darn it, I better have a neural structure that supports the biologically relevant set of stimuli that I'm going to experience in the natural world. If it doesn't, that set of biological structures is not going to survive from generation to generation. But yes, you're right. There is a way that can be constructed. It's just doesn't serve a good purpose for the organism. Yeah.
I think the structure in the middle, again, very middle. Why isn't the structure at the very center?
Why isn't the structure at the very center? What do you mean by the very center? Oh, because there are medial superior olives on both sides of the brainstem. These are neuronal structures in the brainstem. So there's one on the ipsilateral side, there's another, they're on both sides. So there's a duplicate of this that's designed to detect interaural time delays for sounds that would come in from this angle before that angle. And so you've got a dual representation of interaural time delays.
So if you isolate, if you close one of the ears, would it be able to detect it? Like if you just block one ear and the sound is close to the other, would it be able to figure it out?
Not well, no, because it's known from psychophysical experiments in animals and humans that if you fake interaural time delay, if you contrive it, that's where people look and that's where animals look. It is the interaural time delay itself that is the cue that is computed on, and if you muck up the interaural time delay, you can't do sound localization. So that was my stop it alarm, and that's alarming because I'm not ready to stop it. So let me just say quickly, since this relates to the homework, I think you want to hear it, right? So what do these medial superior olivary neurons do? They are coincidence detectors. This system is not going to work if a signal from one side is sufficient to electrically excite and cause a neuron to fire in the medial superior olive. It must be the case that a medial superior olivary neuron fires if and only if it gets coincident inputs from two sides. Then the neuron that fires, and you saw in the movie that as interaural time delay changed, the position of the neuron that fires changes with it. That says there is a place code in the medial superior olive for signaling interaural time delays. Individual neurons have a receptive field that is tuned to a particular range of interaural time delays, and the fundamental computation that must be done is that neuron fires if and only if it receives coincident inputs on its two bipolar neurons, dendrites. Okay? So how can that happen? This is relevant to your homework problem. In the homework problem, I'm going to have you work on this model. You've got a resistor here that represents a soma. Here's one bipolar dendrite, here's another bipolar dendrite. It has a length L. You've got an excitatory input landing here and an excitatory input landing there. And I'm asking you, how can you build an analog AND operator from this neuron system? So what are the constraints specifically on L? What must be true of L for this neuronal system to function as an AND operator? So let's just go through those intuitively. I'm sorry to go long here. The important thing is we're going to assume if this neuron fires when voltage is equal to some threshold value. All right? And you can work out from the homework assignment what the details of that threshold value. So here's the key idea. Synaptic input must be sufficiently far from the soma that one input alone doesn't generate an action potential in that neuron. In other words, a voltage that's above that threshold that I just gave you. Why do we need that? Because it's an AND operator if and only if the neuron will fire given two simultaneous inputs. If it fires on just one, it doesn't qualify as an AND operator. So this sets a condition on L, and it sets a lower bound on L. L must be at least a certain length. It has to be at least of a certain spatial extent to satisfy this condition. However, if the input is too far from the soma, then the two inputs together, arriving simultaneously, don't cause the new neuron to fire. So the bottom line is that the synaptic inputs can't be too close, but they can't be too far. If they're too far, you don't make the neuron fire at all. That's useless. It's certainly not an AND operator. If they're too close, you don't get the AND operation. And so there is a range of L values that set how far and how close these synaptic inputs should be to the soma in order for this AND operator to be implemented. but neurons implement AND operators. It's a logical operation. It's involved in coincidence detection. Coincidence detection is everywhere in many different computations performed by neural systems. And Monday we will see how neurons can compute an AND NOT operator. And what Koch and Pozier, they showed this, and what they implied from doing this is that armed with a neuronal mechanism for building an AND operator and an AND NOT operator, any logical operation can be built as the composition of AND 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 NOT operators. Neurons are capable of performing universal computation. Wow. That sounds pretty good. Universal computation. Wow. That sounds important to me.